Today we are going to study about tryptophan operon. Tryptophan operon is an operon, a group of genes that are used or transcribed together that code for a component for producing of tryptophan. So this is the TRP operon. It is present in many bacteria but was first characterized in E. coli, that is the Escherichia coli. The operon is regulated so that when tryptophan is present in the environment, the gene for the tryptophan synthesis are not expressed. It was an important experimental system for learning about gene regulation, and it is commonly used to teach gene regulation. It was discovered in 1953 by the scientist Jacques Monod and his colleagues. The tryptophan operon in E. coli was first repressible operon to be discovered. While the lac operon can be activated by chemical allolactose, the tryptophan operon is inhibited by the chemical tryptophan. The operon contains 5 structural genes: tryptophan E, tryptophan D, tryptophan C, tryptophan B and tryptophan A, which encode the tryptophan synthesis. It also contains a promoter where RNA polymerase bind to the receptor gene, that is the tryptophan R, which further synthesize a specific protein. The protein that is synthesized by tryptophan R then bind to the operon, which then cause the transcription to be blocked. In the lac operon, allolactose bind to the receptor protein, allowing gene transcription, while the tryptophan operon, that is the tryptophan, bind to the receptor protein, effectively blocking gene transcription. In both the situation, repression is that of the RNA polymerase transcription of this RNA polymerase transcribing the gene in the operon. Also, unlike the lac operon, the tryptophan operon contains a leader peptide and an attenuator sequence which allow for the graded regulation. It is an example of repressible negative regulation of the gene expression. Within the operon regulatory sequences, the operon is blocked by the repressor protein in the presence of tryptophan, which prevents the transcription and is liberated in the tryptophan's absence, thereby allowing transcription. The process of attenuation complements the regulatory action. The operon operates by a negative repressible feedback mechanism. So in repression we are going to know the repressor for the tryptophan operon. It is produced the upstream by the tryptophan R gene, which is constitutively expressed at a lower level. Synthesized tryptophan R monomer associated into the tetramer. These tetramers are inactive and are dissolved in the nucleoplasm. When tryptophan is present, these tryptophan repressor tetramer bind to the tryptophan and cause a change in the repressor conformation, allowing the repressor to bind to the operon. This prevents RNA polymerase from binding to and transcribing the operon, so the tryptophan is not produced from its precursor. When tryptophan is not present, the repressor is in inactive conformation and cannot bind the operon region, so transcription is not inhibited by the repressor. Then attenuation, it is the second mechanism of negative feedback loop in the tryptophan loop, and the repression system target the intercellular tryptophan concentration, where the attenuation responds to the concentration of the charged tryptophan RNA trap, and thus this tRNA trap repressor decreases the gene expression by altering the initiation of the transcription, while attenuation does not be so altered by the process of transcription that's already in the progress. This TRPR repressor decreases the transcription by the factor of 70 attenuation and can further be decreased it by a factor of 10, thus allowing accumulation repression of about 700-fold. Attenuation is made possible by the fact that in prokaryotics, the ribosome began translating the mRNA, while RNA polymerase is still transcribing the DNA sequence. This allows the process of translation to affect transcription of the operon directly. At the beginning of transcribed gene of the tryptophan operon is a sequence of at least 130 nucleotide termed the leader transcript, that is the transcript TRPL. That is, at the beginning of the transcribed gene of the tryptophan operon is a sequence of an at least 130 nucleotide termed the leader transcription, that is the tryptophan L. Lee and Yonovsky in 1977 found that the attenuation efficiency is correlated with the stability of the secondary structure embedded in the tryptophan L, and the two consequent hairpin of the terminator structure were later elucidated by Oxender et al. In 1979. The transcript include four short sequences designed in 1 to 4, each of which is partially complementary to the next one. Thus, three distinct secondary structure hairpin can form 1 to 2, 2 to 3 or 3 to 4. The hybridization of the sequence 1 and 2 to form 1 and 2 structure is rare, because the RNA polymerase wait for a ribosome to attach before continuing the translation past sequence 1. It would prevent the formation of 2 to 3 structure. The formation of the hairpin loop between sequence 2 to 3 prevent the formation of the hairpin loops between both 1 and 2 and 3 and 4. The 3 and 4 structure is transcription termination sequence, abundant in G and C and immediately followed by several uracil residue. Once it forms, the RNA polymerase will dissociate from the DNA and transcription of the structural gene of the operon cannot. The functional importance of the second hairpin for the transcriptional termination is illustrated by the reduced transcription termination frequency. Observe experimental destabilizing the center G plus C pairing of this hairpin. Part of the leader transcribed code for a short polypeptide of 14 amino acid termed the leader peptide. This peptide contains two adjacent tryptophan residue, which is usually, since tryptophan is fairly uncommon amino acid. About 1 in 100 residue in a typical E. coli protein is tryptophan. The stand 1 in the tryptophan L comprises the region of encoding the trailing residue of the leader peptide. Arginine, threonine, serine conversion is observed in these 5 codon, whereas mutating the upstream codon does not alter the operon expression. If the ribosome attend to translate this peptide while tryptophan level in the cell are low, it will stall at either of the two tryptophan codon. While it is stalled, the ribosome physically shield sequence 1 of the transcript, preventing the formation of the 1 and 2 secondary structure. Sequence 2 is then free to hybridize, while sequence 3 to form 2 and 3 structure, which then prevent the formation of 3 to 4 termination hairpin, which is why the 2 and 3 structure it is called anti-termination hairpin. In the presence of 2 and 3 structure, RNA polymerase is free to continue transcribing the operon. Mutational analysis and studies involving the complementary oligonucleotide demonstrated that the stability of the 2 and 3 structure corresponds operon expression level, and if the tryptophan level in the cell are high, the ribosome will translate the entire leader peptide without interrupting and will only stale during translation termination at the stop codon. At this point, the ribosome physically shield both sequence 1 and 2. Sequence 3 and 4 are thus free to form the 3 and 4 structure, which terminates transcription. This terminator structure form when no ribosome stall in the vicinity of the tryptophan tandem, that is the tryptophan arginine these codons. Either the leader peptide is not translated or the translation procedure smoothly along the stand 1, which abundant charge the tRNA tryptophan. Moreover, if we see the ribosome, it is proposed to the only block about 10 NTS downstream, and thus the ribosome staling in either the upstream and the glycine. So, further downstream, threonine do not seem to affect the formation of termination hairpin. The end result is that the operon will be transcribed only when tryptophan is unavailable for the ribosome, while the tryptophan L transcribed in consecutive expression. This attenuation mechanism is experimentally supported. First, the translation of the leader peptide and ribosomal staling are directly evident to be necessary for inhibiting the transcription termination. Moreover, mutational analysis destabilizing or disrupting the base pairs of the anti-terminator hairpin result in the increasing termination of several folds, consistent with the attenuation model. This mutation fails to reveal attenuation even with starved tryptophan. In contrast, complementary oligonucleotide targeting strand 1 increase the operon expression by promoting the anti-terminator formation. Furthermore, in histidine operon, compensatory mutation shows that the primary ability of the stand 2 and 3 matters more than the primary sequence in the inhibiting attenuation. The in attenuation where the translating ribosome is stale determine whether the termination hairpin will be formed or not. And in order for the transcribing polymerase to capture the alternative structure, the time scale of the structure modulation must be compared to that of the transcription. To ensure that the ribosome bind and begin translation of the leader transcript immediately forming its synthesis, a pause site exists in the tryptophan L sequence. Upon reaching this site, RNA polymerase pauses the transcription and apparently waits for translation to begin. This mechanism allows for synchronizing of transcription and translation of key element in attenuation. A similar attenuation mechanism regulates the synthesis of histidine, phenylalanine, and threonine. Thanks for watching. Do like, share, and subscribe.